You can get yourself situated at the seats, and as Lisa introduces each of you, the new ones, you can just wave and say, yeah, that's me. That way you got time to settle in, and I can give you some instructions while Lisa does the introductions.
All right. In addition to our speakers that have already presented, we have Dr. Tiwari, who is the Quality Assessment Lead acting in the Division of Process Assessment 3 in the Office of Process and Facilities in the Office of Pharmaceutical Quality. We also have Dr. Tyner, and she is the Associate Director of Science in the Intermediate Office of the Office of Pharmaceutical Quality. Dr. Connor, who is the Director of the Office of Bioequivalence in the Office of Generic Drugs. Please welcome our panel.
As with all our panel, we're going to take questions from the two microphones here in the room, as well as questions that have come in online. Also, I think that some of the panelists, based on what they've heard, conversations, so on and so forth, may have some things that they're going to share. We'll get to those in just a bit. But let's go ahead and start with an online question, if we could, please.
All right. If a company makes a change to their inhaler product, this is a leftover from the last one that we didn't get to. If a company makes a change to their inhaler product after conducting their pivotal PK study or clinical endpoint study, how would the company know what justification to provide for this difference? How would they know whether they need to submit a bridging study or additional data for this difference?
All right, who wants to field that one?
Okay.
I looked at each side of me and I think it goes beyond just bioequivalence, but if you were going to do that, you would pretty much have to put together your thoughts, any data you already have, and determine, first of all, for yourself whether you think, you know, the extent of the change and how significant it is, and put together your arguments for that you already have enough data, that you have a proposal for doing a comparison or bridging between the old and the new, and simply submit that to, if it's already a filed application, to submit that to your application with as much information and as much clear rationale as you can, to see what the judgment of the agency is as far as what they think needs to be done. If your argument is compelling enough, you may you may have a positive result in that the agency reviewers may agree with you. If not, we'll get back to you and say what we think is important to cover this change. Since there's no SUPAC like documents for these products, we're all kind of doing it on close to a case-by-case basis, I think. And so it's really kind of a dialogue between you and the reviewers and the review staff about what really needs to be done for that level of change.
All right. Anybody else? Okay. Microphone number one, nice and close for me, please.
Hi. Thanks again for putting this on for all of us. Question on timelines. Next year, if an application was submitted with priority review, how close would a complex generic achieve a 6-month review time?
I would say I don't really recall that there is a 6-month review time in our competitor, right? So I think that you can have, for a priority application, you can have an 8-month review time if you successfully file correspondence describing the facilities used in that. And I've been very, very good about meeting our commitments. So I think you can expect that if you submit an application, that with a very high probability you will get your complete response by the end of the time period of the appropriate commitment.
Okay, another online question, please.
How open is the agency to accept ANDA applications with PBPK PBPK modeling, virtual BE, or non-Q1, Q2 formulations of different dosage forms, ophthalmic product, sustained release, long-acting injectables, topical products, and OINDPs.
Pretty big question, covers a lot of different things. And so I think you heard today that we're beginning to set up the framework for how you might submit PBPK modeling in a pre-NDA meeting and in an application. So it's certainly, and I think what you should have taken away from today is, if you're trying to justify a different approach, then a modeling approach is one piece of the information that you can use to help make your case. I think you also should have taken away that if I have, potentially I measure a difference in a physical chemical property for a Q1, Q2 formulation, and I want to understand how significant that difference is, that a modeling and simulation approach might be very appropriate to help provide support to an answer for that question. I think you also heard a previous question where someone asked about, well, what if I have a non-Q1, Q2 formulation? I think the answer you probably heard from that is that might be a much more challenging situation to use modeling and simulation approach. And I want to expand on that a little bit so you understand why. So if I have a different formulation, that means I'm exposing the product to a different excipient. Right? Sometimes with different excipients, you're worried about the impact that they have on the pharmacology, on different types of say if it's a skin excipient, right? You might say, well, that might be more irritating than the other one. Sometimes we ask for in vivo data to resolve some of those differences around the differences in the ingredients. When you have a Q1, Q2 formulation, you think about it from a risk-based approach, or as Sam Rainey likes to say, failure mode-based approach. You have much less failure modes when you have a Q1, Q2 formulation. So that's an important distinction to think about when you're proposing novel approaches as to what are the differences between my product and my reference product. The smaller those differences are, the lower the risk is, and you may be more successful with an alternative approach the closer and closer you get. And I think we've tried to embody, through the meeting process, through our product-specific guidances, you can see us moving toward that type of approach. The closer you are, the less risk you see. The farther away you are, the more data you have to provide. And I think that goes also for the modeling and simulation approaches, as well as the full set of bioequivalence and pharmaceutical equivalence data that you need to provide.
Someone mentioned as part of that huge selection of types of products, ophthalmics, and we could also, for this discussion, put in things like injectable, parenteral products. You just have to remember there are certain things in our regulations that are requirements that certain types of products have to have. And I would refer you to 314, I believe it's 94, it could be 93, but where it lays out that certain types of products have certain requirements coming in to even be acceptable for filing. So if you're doing a parenteral product, what we would term loosely as Q1, Q2, with a few exceptions, is a requirement coming in. You will have a great deal of trouble even getting the application filed if you don't follow those regulations. And the same thing, ophthalmics has their own restrictions on how they are composed and what the exceptions are. It's all in those sections. We don't have as much, we have very little freedom to just say, oh, the heck with the regulations. You can come in with something that's very different, because the regulations bind us as well as you as far as requirements. So there are certain products where it isn't really just our choice or the fact that we're too conservative. It's actually a requirement.
All right. Microphone number 2, nice and close for me, please.
Hi. Question on drugs with the potential to be abused. So with the addition or approval of a new generic, is it expected that a new generic would replace what's already on the market, or by increasing accessibility, it could increase the availability of it to be abused?
So, I think your question is, do additional products that can be additional generic products tend to change the potential for abuse? I think FDA has published some work on this, where they've looked at this question from more of our safety and epidemiology office has looked at this question to understand what happens in the opioid marketplace. And what the conclusion there was that that marketplace has really been it's really reached the saturation that everything is generic already, and it's really the generic market take from each other when there's new products approved. So we didn't see any evidence that approving additional generic products and that have any impact on the availability of the non-abuse deterrent products to be, there's a large number of media release opioid products that are on the market already.
All right, I know we've got another online question, please.
During pre-ANDA meeting wait periods, if a sponsor generates more data about the questions or supplement their position, how can they add this information for discussion during pre-ANDA meetings?
Oh, we talked about this earlier during my session. I think the best way, and typically we don't accept additional information or new questions as well during the review cycle, that 120-day period when we're reviewing the material. Should there be something that you feel is critical, I would say reach out to the project manager who will communicate with the review team, but in general, it's not something that we encourage.
The way I would approach that is try to put all of your information up front in the package. And sometimes there may be cases where if you say, well, I have this study that's going to complete in 30 days, or I have this data package that has all of the raw data from my pilot study. You may not want to put that in your package, but what you should put in your package is all of the data that you do have potentially available, so that if someone reviews the package, they can say, oh, I'd like to see that to answer the question. So we really are opposed to sort of adding things in later. So you want to sort of declare the scope of information that's available in that original package as much as you can. This also goes for the other related thing that we've seen that we don't like is we come to the meeting, a few days before the meeting you've gotten your preliminary response. Maybe it's not the response you wanted to hear, but you did happen to have Plan B hanging around, and at the meeting you'd like to talk about Plan B. What we really encourage you to do is in your original submission, outline your Plan A and your Plan B. So then you'll be able to at least get some, because what will happen if you sort of arrive Plan B at the meeting is you'll say, well, we're not able to talk about that, please submit another meeting request. If you lay the agenda out in that package, here are the approaches that we'd like to discuss, then you'll be much more likely to get feedback on which is the most, the approach that's most likely to lead you to success in that particular case. So, I would say don't keep it in your pot, don't keep it hidden. There's no value to you in keeping it hidden. You won't get a different answer if we don't know about it or we do know about it. You'll still get the same answer on your original approach, whether we know that you have an alternative or not. So it's not a sort of framing or strategic question. It's really I think this is how we want to run the meeting process, to be as straightforward and clear as you can, and you get as straightforward and clear advice as we can give you about your different proposals.
Follow up on that a little bit. I think it's important to, I had several people ask me questions during the break that if you're submitting a follow-on meeting request or additional meeting request after you've completed a meeting request, do you get a different type of timeline, a faster timeline? And the answer is no, you don't. You have the same timeline. So this just is more evidence that you should put everything that you can up front in your first, in your initial meeting package.
Microphone number 2, nice and close for me, please.
First, thank you for the nice workshop, which allow the opportunity for us to talk face-to-face. So I have a couple questions, pretty general. First question is for the complex API as a peptide. So as we know, like a lot of ILD, which they have recombinant peptide, and the generic. What people try to do is use synthetic peptide. So our question is, normally in the ILD, we cannot figure out what kind of salt form or acidic form for the API. So is there any like restrictions for the Sesantic peptide? It has to be the salt form or acidic form. If all the like potency and impurity profiles are comparable. So is any restrictions for that? Or any?
Actually, it's very common lot of applications we are receiving, and we have addressed some of the question recently. The impurity profile has to be the same as compared to RLD. Certain things like in recombinant, there are diastereomer things are not there, but synthetic there are some different impurities. So one has to have the profile will be little bit different.
Yes, yeah. As normally synthetic is much pure, less impurities.
Right. Yeah. And there will be different process impurities. So you have to address all those things.
Okay, so basically the form is not so critical, like salt form or
yes.
Form. Okay, thank you so much.
Yeah, I think Bob, go ahead.
So make sure you know that under how the A&D process works, you have to have the same active ingredient, not the same active moiety. Right? Just remind you to set, to clearly.
Yes, as you hope the finished product will be the same.
Right. So just make sure that you're clear on that. That the active ingredient, which includes the salt, has to be the same as the reference product for the generic product.
Oh, okay. Thank you. So for my can I ask another question?
Since there's nobody lining up, you get one more.
Okay, thank you. Okay. The second question is about the auto-injector, the ICG stability orientation. For this pen, as we know, there's no head space, but I also see the guidance. So we have to have minimum 2 orientations. I would like to know like what's the rationale behind, because the matter is horizontal or vertical. Because they're the same. They're in contact in both ends. So yeah, I just maybe just for my own like curiosity.
So for the stability studies, and this would be something that you could come in with a question early on, is to discuss the rationale. I mean, based on the geometry of your device, of the auto injector, and have a rationale and explain why you want to do the stability study one way versus another.
Why we need the 2 orientations, not just the one, since they're the same basically.
All correspondence question before you do the study can get a good answer on that.
Because there's always going to be exceptions, right?
Yeah. So any plan for the 505(b)(2) complex product workshop down the road?
I don't think we have any planned right now.
There are different organizations within FDA who are responsible for the 505(b)(2) program.
Thank you so much. Yeah.
Okay. And I want to give my panelists a warning. We've got some more online questions, but as we wind down, I'm going to be asking each of you for some closing thoughts to share with industry before we finish. And Katherine, I'm going to be putting you on the spot first, so I wanted to give everybody a heads up, and then we'll finish with Robert so he can wrap up. But I know we've got more online questions. Next online question, please.
Okay, we're developing an extended release oral product and have CMC related questions. Can we request a pre-ANDA development meeting?
Short answer is yes.
I love those short answers. Microphone number 1, nice and close, please.
Yeah, I would echo the other question about orientation and the stability studies. For auto-injector or some of the pre-filled syringe, because there's no bubble, so whatever orientation you have, it's going to be the same. So if we identify worst case, do we still expect 2 orientations in the stability studies?
Right, and so again, that'd be something that, because we would judge it based on your application, you'd want to submit it either through a control correspondence, or if you have other questions, then you can go through a PANDA meeting.
Sure.
Pre-ANDA meeting,
sorry. Sure. So for the pre-ANDA meeting, we have also for Q2 question. This Q1, Q2 discussed quite a lot of times. So this question about Q2, so it's a real case study. We have product under development, and during close development, when we analyze doing reverse engineering of the reference standard, what's on the product label actually is different from what we analyzed. In that particular case, as we understand, in order to get the filings, the must-have items to have the Q1, Q2, particularly the Q2 part. So how do you go through this? Is there a mechanism or path forward to address this question?
I think people have tried to ask this question in lots of different ways at this meeting, so I think we should try to address it as clearly as possible. So we do recognize cases where, as you're doing your Q1, Q2 analysis, you may measure something and then you will send it in to a control correspondence at FDA, and you think that you're the same, and you'll get back an answer that you're not the same, right? So then you have to do some analysis on what your next step is. So I would, if I were in your shoes, I would double check my work. But if you're still confident that you send in a formulation that is what you measured from the RLD, I would go back probably first through the control correspondence, but generally in a control correspondence about Q1, Q2, you just provide the formulations and we provide an answer. I would come back with your follow-up on that with not just here's my proposed formulation, but here's the set of experiments that I did to support my belief that what I have is correct. And this may then give FDA some more information to understand whether it is, you're still, you're still missing something, right? Maybe it's a case where there is something in the RLD, but they'll give us more information internally to say, what should we do? Should we look at it in a different way? Should we have some of our laboratory staff look at it? That's something that's happened in some complex product cases where FDA has gone out and measured and said what's really in the RLD product. So so the more information you provide to us helps us internally then determine what type of issue it is. So we encourage you to come back with the data around the cases where you think that it is a difference. But I think you need to want to do your due diligence to try to identify where the level of uncertainty specifically is. In that, is it really, don't just jump to the conclusion that the RLD, FDA's got the wrong idea about the RLD. Really look at, is there a complex excipient in there that's needs more characterization? Is there there's a lot of things that can go on there. So we encourage you to sort of send a follow-up, but really trying to focus on the specific issue. So don't just say, well, here's my formulation again, right? If you do the same thing again, you're going to get the same answer. You've got to do something different. So you've got to provide more information to really identify that there may be a real issue here with the data that you have you've obtained about the RLD. So I think that's the way that you should approach that particular case. And I hope, I know that it does occur, but I think it actually is quite rare. So in most cases, the Q1, Q2 answer you get reflects what's in the reference product. So if there's I think you have to be aware that if you have that case, it's probably unusual. So you want to really make sure that you've done your due diligence before you come back and say, oh, the RLD label is wrong or something like that.
For this actual real case, particularly actually EDTA, and it's possible that RLD product had a few changes post-approval, and during our course submission, there might be labeling change or the compositional change. So I figure there might be a path forward, either through pre-NDA meeting. We're going to put all the analytical package into the meeting package, but still, how to address this continuous change of the reference product.
But that's part of the environment the generic industry has to operate in. You have to be monitoring the reference product for changes in their labeling that can affect your product. Changes in their changes in their product as well, the reformulations that may affect your product as well. That's just part of the risks and things that you have to monitor during your development process.
Thank you.
Okay. Go to our next online question, please.
Will the agency be sensitive to reducing requirements when the RLD is very, very expensive, provided there isn't conclusive evidence it would reduce safety or efficacy, i.e. reducing retention samples of the RLD in a BE study?
We're strictly about retention samples now. There is, I guess, awareness that the retention samples, it's a rather curious thing that when retention samples were originally, the need for retention samples were originally established, And I wasn't there, so I can only guess at what the rationale was. The guidance about how many retained samples you needed at a given site or in a given shipment was, I believe, 5 times release testing. And that's always appeared to be kind of silly to me. Not the total amount, but the rationale for figuring out the amount you need. Because the purpose for having retained samples is not to do release testing again, or several times, or anything. It is really to, as a guard or a check against certain types of fraud. And we have a history, fortunately pretty long ago now, where one or more sponsors made copies of the reference drug that looked like their own and then tested essentially identical products against each other. And this is the attempt to protect against that type of activity. And so what is needed is certainly enough samples retained, and again, to impress it, because we've had some kind of controversy lately, that each separate shipment to the CRO needs to have their own retained samples. But what's really needed is at a bare minimum, we need enough samples to be able to do identity testing. And our lab, our lab does do that testing. In case you ever wonder what happened to your retained samples once the inspectors pick them up, everyone is tested. Every group that's picked up is tested in our lab, usually at St. Louis. But probably for most products, the 5 times release testing is more than is needed to do that function. So we've been trying internally we've had several meetings probably within the last month or so, trying to say, well, what is the correct number? What is the minimum we can get away with and still have this protection and still do these functions and possibly repeat it once or twice in the lab? It usually, for most products, comes out less than 5 times release testing. So we are working very actively on trying to get, to lower the amount, especially for those, for complex, expensive products, to say, we still need to do this, but we don't need that high number. We can establish a number where we can get this done with less samples than what's, specified. And so we're working actively on trying to kind of reduce that burden a little. At some point, you will still have to submit those expensive samples. And we've already made some progress in that, because I think we have a guidance out that addresses MDIs and nasal sprays and things like that are obviously expensive and difficult to manufacture and everything, to make sure that we have the right number to do this. And we're going to probably in the future be working on other categories of products as well, so that it is a much more reasonable request without giving up that protection. So that's about the progress we're in now. We're actively working and discussing what's the really right number for each product, so we can get this done but not overly burden the sponsors.
With the FDA asking about tightening the limits for unknown impurities to 0.1%, is there any other option to justify other than the RLD data and impurity qualification? Is it possible to justify a higher limit based on QSAR qualification if the impurity is not a metabolite?
I think comparison with RLD is the best option you have.
Close-ups on that one? All right. And actually, that's a fine segue, because I would like to make sure that we've got enough time for this. With the folks that are online, you've got an audience about 600 people in industry right now, and what I'm looking for are some, based on everything that was discussed over the last 2 days, just 1 or 2 minutes closing words of wisdom, the recommendations for them to be more successful in their endeavors. I'm going to start with you, Katherine, if you would, please.
All right, so I have 3 recommendations. The first is come talk to us early. The second is when you come talk to us, bring questions, but bring them fairly targeted. Don't just say, is this great? Will this work? Ask the specific questions. And as you ask those questions, the third part of advice that was touched on already in this panel is bring the data. And if you have the data, I mean, there's no benefit to hide it at this point. If you're asking the questions and want to get some answers and get what our thoughts are, show us the data so we can have a really good scientific discussion.
All right, thank you for that. And Kamal, please, your thoughts.
I think I concur in that application is very important. And when you have basically planned a meeting, just try to provide as much information as you can immediately so that there are no surprises. As Bob suggested, it will help. More information you have, it will be beneficial to industry and as well as to the agency.
Okay. I'll focus some of my comments on some. I heard some interesting things today. I've heard them before, and I actually was asked in the hallway this as well. Is comments on the so-called what we've chosen to term the weight of evidence. We've even gotten arguments that that's kind of an inaccurate term, but we're sticking with it for the moment. But a lot of these products, if you look back through our regulatory history, they're all, and the ones I'm talking about are now drug-device combinations. So we have nasal sprays, we have MDIs, we have DPIs, and there are probably some that I'm forgetting. In the early days, we had, there were products that were even in those days available for generic competition. Yet we and the industry and the scientific community could not really figure out a test or an easy way to get these approved. So for a number of products, they just kind of went on year after year with no path to approval. And obviously it frustrated the industry, it frustrated the public who still had to pay the high prices of innovator products, and it frustrated us because we were not in a place where we could approve these complex products. Over time, we sat down and I was on several of the committees, and it probably, even getting to the starting stages of getting approval path for these, took probably 10 years of work with virtually everyone that you see up here and quite a few others trying to say, well, what do we really need? What would be convincing to us and to the public with these very complex products? It's not just doing PK on a oral product, because that kind of answers all your questions. We don't have a single test or even two tests that answer all our questions, about this product and how similar it is to the original. So we developed this kind of, I called it in the early days a shotgun approach. We just said, well, what are all the critical parameters? What are all the things you would want to know? What are the things that could go wrong? What are the failure modes as far as judging bioequivalence for these products? Because there's the device, and there's a whole, if it's a device that aerosolizes or delivers the product in a certain way, there's actually several components that could be, could go wrong or be designed differently so it didn't perform. There's the drug itself. And there, there's the kind of clinical use. How does this all come together? So we essentially took everything we knew that could possibly go wrong, and we put it into one big package. So what you see is this weight of evidence approach, which there is no single test that'll give you the bioequivalence answer. You have to look at all of the components, and sometimes at various life stages and things like that. So you end up with this seemingly large group of tests, each one of which usually has a statistical test on it that you could possibly fail. And when we got to that stage of saying, we have this large group of tests and a large group of areas on different components of the product, and when you get to a certain point, you ask yourself, wow, I've done a lot, and what else could go wrong? I've hit every single thing I can think of. And I've tested everything, and each one of them, in a very challenging set of tests, by the way, I don't need to tell you that, it matches everything we can think of. And so that's the weight of evidence approach. No single part of that can stand by itself. So the questions about, hey, can you just let me do particle size, aerosolization, and just nothing else? Well, no, you can't, because that only answers one small part of the question. Could I just do PK and just forget about everything else? Well, at this stage, no. Because it doesn't answer all the questions. So this weight of evidence is trying to say, we couldn't, in our current way of thinking, unless, perhaps in the future it'll be different, we can't really have a convincing answer or judgment without looking at all the parts. And there's no single test that will give us that the way it does for simple tablets. And so that's really the explanation. It seems like a lot, but without that progression, without that set of tests, we would have no path to approval for these products. Because I don't really think there exists that single perfect test that will give you the whole answers and give you, us, and the public confidence. So that's the reason why we do it. And it's really, as onerous as it seems, it's better to have a path to approval and to be able to get an approval than just be left with nothing. With no way to get these approved at all, and to have the public going on decade after decade with no generics in these products. That's kind of the history and the rationale for why this weight of evidence approach was thought up and developed, is that if you go through that gauntlet of tests and reach the end, I don't think there's little to no doubt that you have an equivalent product. Seems like a lot. It's pretty expensive, pretty difficult, but at least you have a path where you can get an approval on this product that you develop.
All right, thank you. And Kris, you were one of our earliest speakers starting. We've come whole circle based on everything that you led us off with and what we've discussed since. Some closing thoughts for you for industry.
Sure. So my comments are going to be centered around the pre-ANDA space, and I think my biggest recommendation would be to read the guidances that we have available for pre-ANDA meetings and for controlled correspondence, and to think about which pathway is the best for your particular questions and what you're looking for, and to read those guidances before you submit either control or a meeting request if you haven't done that yet.
Okay. And Robert, before you go into your tips and tricks, but you're also segueing in. You get to tie up all of the 2 days. And so if you can go ahead and do that, go ahead and hit that microphone there.
So thank you, Jeff. So it's my pleasure to be able to close this meeting. I think it was a fantastic meeting. Do people agree? I hear the applause in the room. I hear the internet. That's great. I think you've heard from our colleagues, right? That you. That's what you should say. Okay. So I encourage you to provide them feedback to this programming, and also say, you want more programming like this. That makes us more likely that we'll be able to do this again in the future. I mean, I think the attendance at this, both in person and online, was fantastic. I think that shows the high interest in complex generics. I think at FDA now, the standard of interest is also that the commissioner retweeted some of our tweets about this meeting. So that's the stamp of approval at the FDA now. So I think that happened too. Again, shows the high interest this meeting has across the organization. And I think here, you got to hear about content across the breadth of different dosage forms and complex products. I think that's a fantastic opportunity. It shows how broad the impact of complex generics is. Talk about complex APIs, ophthalmic, dermatological, inhalation, nasal products, long-acting injectables, abuse deterrent formulations. Here, and if you think about it from the business point of view of the generic industry, there's still billions of dollars of products in there without generic competition. That's one of the reasons for the high interest in this type meeting, is to bring access to generic versions of that. So at FDA, we can talk about it, but you in the industry, as Cook said in her opening remarks, there won't be any generics unless you give us applications to review. So, there's a big part for the industry in providing access to this product. And hopefully through the GDUFA 2 pre-ANDA system, you now have a better and more predictable ways to interact with FDA around these complex products, to move toward developing your product, submitting your application, getting your application approved. That's really the goal of this type of workshop. You know, we recognize that in some of the product categories, they're very far from ANDA submission. Some of the other product categories, the transdermal products, FDA has been approving many products. You heard a lot more about deficiencies from the review process and things that our reviewers have seen, depending on the particular product categories or in different areas and spaces. But again, the overall goal here is to move these complex products through development, submitting applications, getting those applications approved. And we hope that this meeting has been very helpful to you in the industry to do that more effectively, to understand the scientific foundations of what we do, understand how to use our pre-ANDA system more effectively. And you know, so if we've done that, I'll feel this has been very successful. And so finally, I just want to thank the people that are responsible for this, especially our SBIA colleagues, especially Brenda, who is a lead organizer on here, and all the staff that worked with here on the logistics, the marketing of this, preparing the agendas, securing the space, making sure everything ran smoothly. I'd like to recognize the speakers. You heard from speakers across our organization, from our super office directors down to reviewer staff. All of these people are very busy, took time out from their daily work to come here, to prepare their talks, to practice their talks, to hone their message, to best communicate to you. So really we want to give all of our speakers a round of applause to thank them for making this meeting a success. And so finally, I just want to wish everyone safe travels home, and we look forward to seeing you at this meeting in the future. Thank you very much.